Alright, tonight's little lesson here is about taking a look at what happens when we start to count the number of chromosomes that are in a cell and some of the things that can happen when those chromosome numbers get off a little bit. This all has to do with genetics. It all has to do with meiosis. So you can see there's a nice picture here. It's kind of cool the way they've color-coded the different chromosomes that are on here, and you can kind of see them sorted out. Two different color patterns, but the same sets of chromosomes separated out into their pairs. So let's take a look how this works. So, first of all, if we catch a chromosome kind of in the act, or sorry to say, if we catch a cell in the act of dividing, when we've got the chromosomes all into their sister chromatids like this, we can actually do what's called a chromosome squash. We can take the cell, or the cells, drop them on a slide, squish them down flat, kind of pop their little cell parts out, and we can get these lovely little pictures of all the little chromosomes spread out like this. So it's called a chromosome squash or chromosome spread. And when you take a look at this, you should be able to see that there's a lot of different shapes and sizes. These are human chromosomes, and there's a bunch of different kinds and shapes of chromosomes that are out there. So we've got some chromosomes like this one right here that are really quite long. Here's another one that's probably its partner, also quite long. And then we have some other ones, kind of like this one, sort of medium size, but notice how that centromere is in a little bit of a different spot, not quite in the middle. And then we have some little tiny ones like this one and this one, quite a bit smaller than the other ones that are out there. None of these are abnormal. These are all really normal kinds of chromosomes, but there's lots of different sizes and shapes that are out there. And as soon as people realized that those were there, they started looking at, well, can we arrange them? Can we partner them up together? Turns out that the partnering system that's used is called the Denver system. It was agreed upon by a bunch of biologists at a conference here in Denver, I think in 1960 or 1961, something like that. And so the Denver system of chromosome classification is kind of what you'd think. You just sort them out according to what the pairs look like. And so here you can see sort of a series of chromosomes all sorted out. They're sorted into some different groups here. You can see like the A group over here. These are all really tall, really long chromosomes with the centromeres kind of right in the middle. You can see the B group over here, sort of medium size, but you can see their centromeres are asymmetrical, up kind of near the top. They're not evenly placed. Then you've got this C bunch down in the middle, quite a big bunch. And these are a little bit on the smaller side, and most of them have the centromeres fairly close to the center. More centered in 6 and then less centered in 12, but they're kind of the medium-sized ones. Chromosome group D here is some of the short ones, and they have a really weird centromere right up at the top. E is short as well, with centromeres right in the middle. And then F and G are the tiny ones, where you've got little bitty chromosomes either in the middle, or little bitty chromosomes with the centromeres in the middle, or centromeres up near the top. And then you've got the last weird pair. These are the sex chromosomes. In this case, it would be a boy because there's an X and a Y. So all those other chromosomes, pairs 1 through 22, are called autosomes, the regular body chromosomes. And obviously this last pair down here, those are your sex chromosomes. So again, this is the Denver system of classification. There's another style that we can use too that's based upon staining the chromosomes, and this can be really useful for cells that maybe aren't in mitosis, where they don't have the sister chromatids set out. And you can kind of see there's these little stripes, little bands of color on each one of them, and you can actually match up the bands. There's a kind of a good picture on the next one here that I'll show you. But again, this is an actual photograph of stained chromosomes sort of sorted out, and they're all kind of crinkled up like this, all wrinkled, because that's just how they splattered onto the cell, onto the slide when they took a picture of it. So here is a better picture of kind of they call it an ideogram, little cartoons basically to diagram this banding pattern. And this dashed line right here, that's supposed to indicate where the centromere would be. So again, it's a little bit harder to see where the centromere is if they're not in that duplicated phase, but it's easier to see the banding pattern. So it's kind of a plus and minus. So just two different ways of being able to sort these things out into this kind of series of pairs, either using the banding or using just the overall shape. So what the goal of doing this is to create something called a karyotype. And so here you can see actually two different karyotypes. Here is up at the top the original pictures. So that's kind of up in here, right? You got the original photograph of what that cell squash looked like when it was taken a picture of, and all kind of spread out all over the place. And here you can see they've literally cut and pasted them out to make a match. So here's the two number 1s, here's the two number 2s, two number 3s. This is a boy, right? Because it's got the X and the Y chromosome. Those were left over. Here's a girl, because she's got two X chromosomes. The X chromosomes are pretty long. They would kind of belong up, they're kind of close in size to number 5 or number 6 in terms of where they match the other chromosomes. But so you can see this is a karyotype now. It's nice organized by color, or not color, by size. And then here is sort of that karyotype analysis. Here's the total number of chromosomes, and then there's the sex chromosomes that are present. And again, even the female, total number of chromosomes 46, and the sex chromosomes are XX, and that makes it a normal female. So in class, you're actually going to be asked to do some karyotypes. You'll be asked to do a couple normal ones, kind of like that one. And you're going to be able to use giant magnets. These magnets are pretty big, that you can kind of move around. They're not little tiny pictures at least, but they use this banding pattern. So this is the lab that you'll be able to do to kind of look at a couple normal karyotypes and look at some of these kind of more unusual karyotypes that you'll see here just in a second. So when you're doing karyotypes like this, they started to realize that sometimes weird things happen. This little karyotype that you've got in here, you can actually see the little pieces of paper that they cut out to sort. But this little karyotype shows something unusual. If you take a look at it pretty closely, it doesn't show all of them. I know it doesn't show 1 through 5. But if you take a look at what's on here, right here, something kind of unusual. There's an extra chromosome number 21. This is also a male. Right, an X and a Y. Everything else is normal. Again, I know they didn't show numbers 1 through 5, but there's something unusual here, and this is what we call Down syndrome. The karyotype of a person with Down syndrome shows, and here's what you need to be able to write down, shows 47 chromosomes. This is a male, that's why it's XY. And then we would indicate what's unusual about this karyotype. It's plus an additional number 21 chromosome. And it's this little tiny chromosome is what makes Down syndrome happen. And if you've ever met someone with Down syndrome before, some of my favorite people in the whole wide world, but they definitely have some unique physical challenges that go along with having one extra tiny little chromosome. How does this occur? This is related to the process of meiosis, which you learned about before. And this is a problem in meiosis called nondisjunction. Nondisjunction is when two chromosomes that should separate don't. So over here on the left in this diagram, we've got the normal one over here. In anaphase, you've got each pair of chromosomes is separating, and they should be going to opposite sides. And so we'd end up with this blue circled cell that have 1, 2, 3, 4 chromosomes in it, and our green cell over here would have also 1, 2, 3, 4 chromosomes in it. And that's normal circumstances. Everything's going right. But for some reason, sometimes an extra spindle fiber, you can kind of see I drew one in there, but sometimes an extra spindle fiber gets attached, or something else doesn't move quite right. And we end up with the wrong numbers of chromosomes in the offspring cells. So here, you can see my blue cell is actually missing a chromosome. My green cell has an extra. So my green cell here has 5 chromosomes as opposed to the normal number of 4. It's got an extra one. So if this was Down syndrome, this would be like chromosome number 21, and this individual could have been born from that. By the way, in humans, if you're missing a chromosome 21, you're never born. So not like there's an undown syndrome. There's only a +1 and you get to live. So again, the results of this process called nondisjunction, during metaphase and anaphase, the homologous pairs do not separate, and gametes are either plus or minus a chromosome, so then the new individual that would be formed from that gamete would be plus or minus 1, 2. If this happened in a non-gamete, like a regular body cell, probably your immune system would take care of that cell and that cell would die. It's really critical when it's an egg or a sperm that has this particular condition. So what do you need to be able to know out of this? Non-disjunction disorders like this, like Down syndrome, generally not survivable. There's very, very few cases that we know of where you could be plus or minus a chromosome and still be born at all. A lot of these non-disjunction disorders are fatal extremely early. We're talking within, months of birth. Like this one down here, Edwards syndrome. Usually you don't live to be 2. But a lot of them, they're so, so non-survivable that the parents would probably never even know they were pregnant. It's one of those really, really early termination issues. So these are the 6 that you need to know. You'll have an experience, like I said, to look at these in lab. But the one you're most familiar with is Down syndrome, which is an extra called trisomy, right? An extra number 21. There's another one called Edwards syndrome, which is trisomy 18. It's actually quite a bit less survivable than Down syndrome. That's why you probably haven't heard of it. But these are called autosomal disorders because they happen to one of the regular body chromosomes, not the sex chromosomes. Turns out there's a few more variations of the sex chromosomes that are survivable. For example, Turner syndrome, you have an X but nothing else, just a single X. So you'd be a female, but you'd only have 45 chromosomes. These two in the middle have some extra kind of weirdness. These are both males, Klinefelter syndrome and supermale. The Klinefelters has an extra X, the supermale has an extra Y, and then superfemale would have 3 X's. In fact, superfemale, a lot of times you can't even tell that their phenotype is actually very, very normal. Supermale phenotype is usually pretty normal, but there's some argument about that. Klinefelters and Turners are definitely different, and you can see both of those phenotypes. So again, here's your 6 types of karyotype non-disjunction disorders you'll need to be able to look at, kind of play with a little bit in lab, and I'll see you in class tomorrow.